DNA is the most common specimen collected for future research on clinical trials, and it is the analyte that is most commonly required for collection by regulatory agencies, and so that's why we focused on pharmacogenetics. Pharmacogenetics is also probably in the lead in terms of biomarkers associated with response, but other types of biomarker research are rapidly also coming up, and so I think that collection of correctly collected serum, RNA, proteomic samples are also tissue samples are also going to be important in the future. But today we focused on pharmacogenetics. There's been indecision about whether specimens can be collected and stored for future research that is broad, and so even though it is not completely blanket consent for future research, it is broad consent that would allow us to do research related to drug response, but also to disease markers of response, so that we could fully understand both the patient response to drugs under therapy, but also the patients who were enrolled on the clinical trial as subpopulations start to emerge during late phase development or even in post-marketing activity. Often, subpopulations don't emerge until you treat thousands of patients, and that's when there's a very large effect size, so you can identify a subpopulation of responders. But what we know is that not until millions of patients have been treated do we often collect enough cases of either adverse events or sub-responders to therapy that we can test. And so collection of specimens in late phase trials and in post-marketing activities allows us to build a large enough data set to fully investigate those subpopulations, especially if we collect appropriate clinical data and phenotype information so that we can associate the phenotype clearly with the specimen and the data that we generate from the specimen. So I think industry has some clear action items. I think that we need to start to build trust firstly for how we're collecting, storing, and using specimens that we collect for research. So we need to make transparent what our processes are and how we're putting them in place in order to assure protection of the patient, but also to assure that we're using the specimens responsibly. So I think both individual companies will start to develop internal tools to communicate the specific procedures within the company that relate to specimen collection, but I also think there are cross-industry efforts we will make to establish some best practices, and I think the Industry Pharmacogenomics Working Group, the International Pharmaceutical Privacy Consortia, and ISBR may likely partner to create those best practices. I think the FDA guidance will make significant headway for US-based studies, and the hope is that potentially ICH would pick up the topic and talk about harmonization of at least the approach to collect future use specimens across the United States, Europe, and Japan.